We are here at the HPLC 2025 conference. I'm together with Georg Schuster from Coriolis Pharma. And Georg, you're presenting a poster here. And the data are also partially from a collaboration that our companies had together. This is why a Thermo Fisher logo is on it. So what's the poster about? And why do you think this topic is relevant nowadays?
Hi, Frank. Thank you. So at Coriolis, we've been helping our clients since more than a decade now with formulation development of biopharmaceuticals. In that field, surfactants play a critical role, and Coriolis has been working on charged aerosol detection and surfactant analysis and quantification since that time. So therefore, we don't want to stop with our development, but also continue in improving our services and analysis.
This is great, and of course we appreciate this. So if we go a bit further into the technical approach, so how did you start this research? What triggered it? So the plan, the experimental plan and these things, what was behind that?
So a typical approach in the community or in the field is to use a single peak method that excludes protein and then elutes the surfactant in one peak for quantitation. However, at Coriolis, we believe a lot in using gradient methods, so separating the surfactant to quantify or to characterize the content and the surfactant in the product. So where you have the benefit of online elution with a single peak and direct analysis, for gradients, you still need to use some offline sample preparation like SPE or solvent precipitation. So we wanted to involve in that step to have the combination of both, using online removal of the protein, but still have the benefit of a gradient separation that gives you also information on degradation of your surfactant.
Mm-hmm. Was there anything in your results that surprised you? What is a key insight actually to communicate?
Yeah, it's actually not so easy to tackle that. So it took us a bit to work that out. We benefited really a lot on the 2D setup, and for us it was important to actually use a setup that you can buy as is and optimize it. But we had to look a lot into column chemistries, and we actually found that the best approach for us is to use a three-column approach. So where unusual columns or usual methods, you use one of an anion exchange to exclude the protein and trap the surfactant. We observed that still residual protein is kept on in the system. So we decided to use a second dimension for trapping that captures the protein that was still in the sample flow path and then elute through both columns to a reverse phase column. So that was very
impressive.
Yeah.
It's impressive. So would you say the results that you generated is something that other pharma companies or CROs could readily use?
Yeah, I think the benefit and what really astonished us as well is the broad dynamic range that we saw on the protein exclusion. So on the poster here we show that we could use, so we used several commercially available antibodies ranging from 20 to 180 mg per ml. And they could have been used, or we could analyze the samples without any pre-treatment, any sample prep. And we were in a fairly good recovery rate of up to 90 to 110%, which is the usual accepted recovery rate in the field, and without even using the polysorbate that is in the product. So that was quite astonishing and really helpful for the whole community working on surfactants.
That's good news. Thank you very much for your time, Georg.
Thank you, Frank.